Next topic is translation. So as we discussed about transcription, we are going to discuss about the same translation. Now, translation is transferring, correct? Now, what is translation? The process where the sequence of the codons on the messenger RNA are read or they are being used and the amino acids so formed are linked with each other to form a long polypeptide chain is generally called as translation, right? So you know very well the sequences of the codon present on the messenger RNA, right? They will be read, they will be utilized, and similarly the amino acids will be formed and those amino acids will link with each other to form a long polypeptide chain. That process is called translation. Now when you talk about the translation, for the translation, first two important processes are first activation of amino acid, correct, and formation of amino acid transfer RNA complex, right? Second one is formation of a polypeptide chain. So let's talk about the first process, that is activation of amino acid and formation of transfer RNA complex. With the help of the enzyme amino acyl transfer RNA synthetase. With the help of the enzyme amino acyl transfer RNA synthetase, right, the amino acid generally goes and gets binded to the transfer RNA molecule at the 3' end or CCA end to form transfer RNA amino acid transfer RNA complex. So what is the name of the enzyme? Amino acyl transfer RNA synthetase, correct? So with the help of this enzyme amino acyl transfer RNA synthetase, amino acids are generally transferred or amino acids are generally attached to the 3' end or CCA end of the transfer RNA molecule to form amino acid transfer RNA complex. Second, formation of a polypeptide chain. For the formation of the polypeptide chain, you require 30S, correct? 30S kind of a ribosome and 50S subunit of ribosome. 30S and 50S type of a ribosome is required in a prokaryotic cell. Okay, you require amino acid transfer RNA complex and you also require ATP and GTP as a source of energy. Okay, so now we will start the 3 steps of translation. Similar step, that is first initiation, second elongation, third termination. So first step, initiation. The initiation step is generally brought about by the binding of RNA molecule on a small 30 subunit of ribosome. So what happens here? The ribosome, right? You have ribosome 30S and 50S. So initiation takes place by binding of RNA, right? Binding of a RNA on the small subunit of 30 subunit of ribosome, right? The initiation codon present at the 5' end is AUG. So the amino acid transfer RNA complex which has been formed, correct, will go and bind to the initiation codon AUG. Now this initiation codon AUG is specified by the UAC anticodon of transfer RNA. So we all know that codon is present on the messenger RNA and anticodon is present on the transfer RNA. Okay, now what happens is that once the amino acid transfer RNA complex goes and binds to what? Goes and, correct now, amino acid transfer RNA complex will generally go and bind to the AUG codon of messenger RNA and this will be specified by the USG anticodon of the transfer RNA. Now the smaller subunit 30S, the larger subunit 50S will join with each other to form a 70S type of a ribosome. Now in prokaryote, that is 70S type of ribosome, the ribosome has mainly 3 important sites, very important. That is first amino acyl A site, second peptidyl P site, and third is exit site. Now the amino acid transfer RNA molecule which has been formed will directly go and bind to the peptidyl P site, whereas the other incoming transfer RNA complexes will first go and bind to the A amino acyl site and are further shifted to the peptidyl or the P site. So the first amino acid, right, the first amino acid, that is amino acid 1 transfer RNA complex, transfer RNA complex. This will go and bind to the peptidyl site, whereas the second amino acid 2 transfer RNA complex will further go and bind to the amino acyl A site and then shift it to the P site, that is peptidyl site. Now, the polypeptide chain which is generally released is always released from the P side. This is generally called first step, that is initiation. Correct? RNA molecule will go and bind to the small 30S subunit of your ribosome. Correct? Now, what will happen? The initiation codon is AUG. Amino acid 1 transfer RNA complex will go and bind to the AUG. Codon of messenger RNA will be specified by the UAC anticodon of transfer RNA. 30 subunit smaller, 50 subunit larger will unite with each other to form 70S type of ribosome. Ribosome has 3 sites, that is A site, amino acyl, peptidyl P site, and exit E site. Okay, now second step is called elongation. The elongation is generally brought about by the formation of peptide bond or the peptide linkage between the two successive amino acids, right? And this is under the control or under the favor of an enzyme, peptidyl transferase enzyme. So peptidyl transferase enzyme will help in the formation of a polypeptide chain, that is formation of a peptide bond or a peptide linkage in between the successive amino acid. Due to the complementary nature of your codon and anticodon, amino acids are placed at the proper position. Now what happens in the elongation is that the ribosome, so example, this is ribosome, right, and this is your RNA. So ribosome, the ribosome will start moving from the start codon to the stop codon. Start codon is initiation codon, stop codon is termination codon, 5' and 3'. So the movement of the ribosome from the start codon to the stop codon of the messenger RNA is called as translocation. That is called elongation. Third step is called termination. Now what will happen when your when your ribosome reaches to the terminator site, that is at the 3' and the termination occurs, right? Now to identify the termination codon in the releasing polypeptide chain, we require R1, R2 and S as the releasing factor. So R1, R2 and S are the releasing factor which are required in identifying the termination codon and also in releasing the peptide chain, right? Okay, now once the termination takes place, the 30S smaller subunit and the 50S larger subunit separate from each other. This is your 30S and this is your 50S. This is smaller subunit, this is larger subunit. Now 30S will get separated from your what? It will get separated from the larger subunit 50S. This is called termination taking place in translation. So translation also requires 3 steps: initiation, elongation and termination, right? Termination will only takes place when the ribosome, that is 30S ribosome and 30S subunit and 50S subunit of ribosome will split with each other. They will get separate from each other and the translation will takes place. Okay, thank you.